Great pleasure to introduce Dr. Jane Stewart. She's an associate professor at Colorado State University. She teaches integrated tree health, and her research interests include kind of a myriad of forest health interactions, including understanding the biology, ecology, and genetics of emerging tree and plant pathogenic fungi using population genetics and genomic methods, studying the interactions between plants and fungi that govern variation in host specialization, pathogenicity, and virulence, and understanding genomic drivers of fungal species delimitation and divergence with a focus on pathogens important to Colorado's forests, shade and fruit trees, and forests worldwide. So without further ado, I'd like to introduce Dr. Jane Stewart.
Great. Thank you very much, Becky. Can you guys see everything in presentation mode now?
Yes, looks good.
Okay, excellent. So thank you very much for the introduction. It's kind of interesting because today I'm not going to be talking about a fungus, which I primarily work in fungal pathosystems, when I'm going to talk about a nematode. So as Becky mentioned, I'm a tree pathologist, and so that allows me to work in forest tree systems, urban tree systems, and I also work in tree fruit. And this is a pathogen that I'm going to talk about that really spans our urban forest as well as now our native forest, which hadn't done so in the past. And so I'll highlight that. This tree that I'm showing here, let me go ahead and start can you guys see my pointer? Just moving around.
Yes, we can.
Okay, good. Okay, so this tree here is an important tree because it was the first diagnosis of a pathogen into Fort Collins, into the region where we live in Fort Collins, Colorado, in 2006. And so what I'm going to talk about today really spans urban trees. So this is an Austrian pine, an exotic Austrian pine that's planted pretty extensively in Fort Collins in our region. About 40% of the trees in our area are pine, and the largest proportion of those are Austrian pine and Scotch pine, which are exotics. And what I'm going to talk about today is basically the movement of a species, a pathogen from these Austrian pine into our native ponderosa pine. And this really spans the overall One Health system or, environment. This is the logo for CSU, One Health, because urban trees are so important to our human well-being, but yet they can also be drivers of potential decline in our forest ecosystems. And so I'm going to talk about a pathosystem that I think we're seeing relationships between what was planted in our urban forest and the movement of pathogen into our ecosystems. Okay, so just to kind of give you the brief overview of health benefits of urban trees. So there was a really nice study done by Wolf et al. In 2020, just recently, where they basically did a meta-analysis of 201 studies looking at how trees improve overall health conditions for humans. And the benefits are numerous. This was focused primarily on urban trees, but the same benefits you would see in our forest ecosystems as well. Some of these are, reducing pollutants and respiratory condition. So 10 of the studies had a positive association with urban trees, decreases in crime. So these benefits can be really wide spanning as well. Mental health and anxiety improvement, immune system improvement, and cardiovascular function improvement. So urban forests are really key to keeping humans healthy in the urban environment, for sure. Another study looked at the estimated removal of air pollutants by trees, and this was looking at all different types of pollutants. The dark areas basically highlight the amount of pollution that was removed in different counties. And this was either 4 to 8 tons per square kilometer. And what you can see is that in areas of the East Coast and the West Coast, where we have more trees that grow pretty much more frequently, we do see a lot of removal of pollution. But this sort of decreases, except for in a pocket here in Colorado. And part of that is because of the urban trees and because of our relationship, I think, to urban to forest ecosystems, which we call the WUI site, so wildland interfaces. So we know that this urban connection is really important, right? However, urban trees can be a bridge for introduced species. And what we see is that basically these urban trees, you might have the accidental introduction of exotic pathogens or pests, and then these can spread into our natural forest. And part of the reasons for these are that we get often exotic tree species that are planted. So these exotic trees may be carrying pests or pathogens that they are not, that they have more resistance to. But as these pathogens move into our natural ecosystems, our native tree species don't have the same resistance. Another reason is because many times in our urban areas, trees are stressed, and so they're predisposed to attack. So there's anthropogenic disturbances that can happen, drought, air quality, just really stressful conditions when they're surrounded by concrete and not in a natural system. And so these trees can be predisposed to different insects and pathogens. And then also tree species are often planted in high density. Now I mentioned in our pine forest, in our urban forest, pines are basically 40%. So when we have something that moves into an urban setting, because we have such a high percentage of the trees that are the same species, pathogens can proliferate and then move into natural ecosystems. And so there's several examples of these. So the steps that we typically see for invasive species introductions include basically first the prevention, right? So the ways that we can prevent is through basically inspections that can occur, but then we might, even these early surveillance techniques may help prevent, but the pathogen's still able to move in. A lot of times early colonization will happen on asymptomatic host tissues. So we have latent fungal disease pathogens or other diseases that occur, but we don't know that they're really associated with the host until the host gets stressed. Some other factor sort of spreads, allows the pathogen to proliferate. And then by that point, we have an outbreak. And so management after we have spread into larger areas is really, really difficult. And there's a huge economic cost to these invasive species. So this graph is showing basically the area occupied, but a non-native species. This is the different phase where we go from absent to pervasive. And then this is some of the example policies we can utilize, and then who has to basically borne the cost of those if an invasive comes in. So, you know, the policies that we have for invasive pathogens before they arrive into an area is basically pre-importation and different inspection measures, which are really vital. But imagine the amount of material that's moving into new areas. The inspections can't happen on everything, and so things get missed through that. So if we have localized spread of a pathogen that's not yet established, but it start becoming established, we usually can set up quarantine, surveillance, or eradication efforts. And eradication efforts are costly, but if the disease or the invasive pest or pathogen gets moved into an area, the cost increases substantially, as you can see here. And at some point, once the disease is pervasive, we adapt. We have to plant other species. Our forest might change. The cost is much higher than it is at these lower levels. So there's some great examples that Papp et al. In 2017 reviewed, looking at this urban natural ecosystem bridge of invasive pathogens. A couple of examples, there's others, but a couple of examples would be chestnut blight. Chestnut blight was brought in on a live plant, so on a Chinese chestnut, in the New York Zoo in about the 1900s. And it moved from the plants into the native ecosystems and basically has changed the face of American forests. Those of you from the East Coast, this is in Tennessee, so many of you know the American chestnut story, and it's one that's really important because it basically, chestnut was removed from our ecosystems and other species have had to come in and sort of occupy that niche. Another example is white pine blisterus. This is an invasive species that's really changed the face of our Western forests, but it was also introduced into the East Coast. And the pathway that brought this in was basically live plants. Another example is Phytophthora ramorum. This is caused by sudden oak death, or the cause Phytophthora morum causes sudden oak death. And this was an invasive species that was brought into the West Coast through live plants. And because it's got such a broad host range, so it's able to impact over 100 different hosts, it's caused detrimental mortality to oaks and other species in California. And so I'll kind of talk about Phytophthora again in a second. And a couple of other examples would be ash dieback, which is a huge issue in Europe. Again, it was imported on nursery stock, and then also Dutch elm disease, which was brought in by wood packing materials, and really changed the face of American cities in the United States and in elsewhere, Europe and Canada. So these Phytophthora diseases are a really great example of how we might have movement from our urban areas into our natural forest ecosystems. So these are just some pictures showing sudden oak death. So the mortality that we see on basically the symptoms that we see on rhododendron and other plant species might be a leaf blight that you can see here. But when this leaf blight gets moved in on tan oaks, what we see is mortality. So each of these arrows represents a tree that has died in California because of sudden oak death. But interestingly, the more research that's done on Phytophthora diseases, what we find is that there's been much more movement of novel Phytophthora species from restoration plantings, basically going from the nursery to a restoration planting in the natural ecosystem. And then those diseases that are in those restoration plantings are able to move into our natural ecosystems. So in a study recently, it's been estimated that about 50 Phytophthora species have been identified in plant nurseries, and 25% of the plants tested were positive for at least one Phytophthora species. And what this means is that when plants are in the nursery themselves, they're already infected with Phytophthora. And if we aren't able to capture that and to identify, then what can happen is that these infected plants, though they may be asymptomatic in the greenhouse or in the nursery, once they're outplanted, they become symptomatic, the spores can spread. And so there are excellent pathways for new introductions of our invasive species. So it sort of branches this pattern where we have something in the nursery, it gets outplanted, maybe in our urban setting, maybe in restoration plants, and then it gets into our natural ecosystems. So I'm going to tell you a story about what we were seeing in Colorado and sort of how this pattern we think has developed in Colorado. So what we were seeing was basically mortality in pines. And these are some great examples of the mortality that we are observing. And a lot of these sites are what we call the wildland urban interface. So I'll call them WUI areas. We were seeing mortality in our urban areas, but then also in areas where people live sort of in the front range, which is basically surrounded by ponderosa pine. They're neighborhoods, but there's a lot of forest, a lot of them back up to the natural forest. And so what we are seeing probably since about 2014 was ponderosa pine decline. And so this is really what the symptomatic, where we might have dieback in some of the crown, uneven distribution of dieback, pockets of trees starting to decline. Here you can see an area where we're started to moving into the forest system. And here's another example of a tree. And so basically in 2017, my lab was trying to figure out what was going on. So in collaboration with Ned Tisserat and Bill Jacoby, who were faculty here at CSU before I arrived, we basically cut down trees that we saw mortality in. And then we caged the wood to capture potential vectors, so insect species that might be coming out. We asked forest health managers and urban foresters to basically send samples to us. So we were getting a lot of samples, and we investigated these trees really thoroughly. And what we found was that we saw symptoms and signs of basically blue stain fungi that you can see here, or black stain. And this was actually Ophiostoma minus. So it was known what this species was. And we also saw exit holes from insects. But the other thing we were able to diagnose was pine wilt nematode, which is not a species that we characteristically find in ponderosa pine. And it was associated with the mortality of the trees that we were seeing in the absence of any other primary pathogens. And so this kind of got us worried about what the pattern we might be observing. So let me tell you a little bit more about pinewood nematode. So pinewood nematode is a nematode, as you can see here. It's Drosophilus xiphilis. And it's a really important disease in our exotic pines. It's really important in other areas of the world where it's been introduced. And I'll sort of show you the pattern of that. But it's capable of causing lethal wilting disease in pine hosts. It has been found in other species, but it doesn't proliferate and produce or go through its life cycle in other hosts. Usually the disease occurs when temperatures are above 20 C. And again, it's primarily found in our Austrian, Scots, and Mugo pines, which are the species that are planted in the urban areas. The life cycle, when we have warm conditions, can be completed in about 4 to 5 days. And it increases, or the life, that span quickens when we have higher temperatures, about 3 days at 30 degrees C. And then we also know that it feeds on blue-stain fungi in dying trees. So it's got this interaction that we have the tree, we have the nematode, and we have the fungus. And it's sort of this three-way disease cycle. So monocamus in other areas where it's been found is vectored by monocamus species. And we have two here in Colorado. We have monocamus clamator and monocamus scutulatus. And before the work that we had done, we hadn't documented pinewood nematode in either one of these species. So it was known that we probably have the vector to move it around, but it wasn't yet documented. And then this would be a grub from these beetles. So the disease cycle starts off where you have basically an infective vector that emerges from a tree. Immediately, those beetles were maturation feed. And so basically what will happen is they'll emerge from the tree and then they'll start feeding on the tops of crowns. And typically it's on healthy trees. They don't necessarily target disease trees or weakened stress trees for feeding. And they'll create basically wounding that looks really similar to this. And what can happen is as they're feeding on the tree, they can inoculate the tree with the pinewood nematode. In an exotic pine, mortality typically happens fairly quickly, especially when we're having hot summer days, where the tree will basically within a month turn straw colored. We know then that the nematode is in the tree surviving, and then the nematodes are attracted to basically the pupal chambers of the beetles. So as this tree starts to decline, more beetles will start to go in to actually breed in the tree. And so in this pupal stage, that's when the nematodes actually get into the beetles, and they basically are in the trachea or the ventilation system of the beetles. So that's how they're carried. The nematode, we know too, will basically, these beetles will also carry the fungus. And so in a stage like this, the nematode is feeding on the tree tissues, but then also feeding on the blue stain fungus. And it's thought that the blue stain fungus is important nutrient-wise for them to get into this pupal stage and for development. So the symptoms that we typically see will be the needles are gray-green. It'll then go to straw-colored, and then the trees can collapse very quickly. You might get isolated branch death that might occur. And what we typically see is that for ponderosa, which is a native pine, we see more isolated branch patterns than we do for our exotic pines. But in an exotic pine, mortality can happen and symptoms can happen in less than 2 months. So these Austrian pine, Scotch pine are super common in urban forestries and in windbreaks, pretty much all throughout the West and in the mid the areas like Kansas and Missouri. But what happens is that where these are planted, when they get infected, there's wilting that occurs. And so basically the nematodes are able to feed in the xylem tissue of the pines, and they basically disrupt the transport of water in these trees. And so you're basically getting a droughting effect. And this was highlighted really nicely in a study looking at 3-year-old Japanese black pine, where this tree was basically inoculated, and then there were actual images showing MRI basically of the lateral movement of the nematode and basically the blockage that's happening, the embolism in the tree. And so you can see that this study first started in July of 15, and then 15 days later, basically that tree has been completely shut off from all of its water. And so this is a very virulent pathogen in exotic pine species. Now, one thing that's interesting about pine wilt is that it is native to the United States, and we know that it's native and it's been here for a long time. And we know this because it had been inadvertently introduced into Japan in the early 1900s and caused pretty widespread mortality in forests in Japan. It was subsequently then introduced into China. There were probably two routes of introduction, one into China from the United States and an introduction from Japan. And then it's also been introduced into Europe. It arrived in Portugal in 1999 and now spread into Spain. But the mortality that they see there is very extensive. So these are some pictures showing the mortality where up to 60% mortality can happen in these Asian forests, as you can see from these photos. So what we think is that the native range of pinewood nematode is the East Coast, sort of where most of you guys are. And it's normally, you can see here, it's outlined by red. And the normal vectors are Monocamus carolensis and Monocamus titular. And we think that the native hosts were really longleaf pine and loblolly. We do know that it has been found in lake states up here. And then in 1979, it was reported causing mortality in Missouri and Kansas. And so in the 1980s, there was a lot of research done in the United States on pinewood nematode. But one thing that's really interesting is it was always assumed that it's in our Western forest, but we're not really sure of that because there's very few reports of having pinewood nematode in our Western forests. So interestingly, there's been this sort of westward expansion or movement across Kansas of pinewood nematode. So this is looking at the movement of pinewood nematode from 1979 to 2018. And the pink counties are basically where we find pinewood nematode pretty commonly. The yellow is where it's been detected, but we're not really sure that it's established. And the white are where pinewood nematode hasn't yet been found. And so there is this idea that the spread of this, it has been happening westward, and then it may not actually be present. And some evidence that we have to suggest is that pinewood nematode was first reported in Colorado in 2006 in the areas in red. So this is Larimer County, which is where I am, and then Weld County. And we've seen an expansion in our urban environments over time, so that it was found in 2011 in Denver County here. And then in 2012, it expanded westward and has been found in multiple other sites. And then it was first reported in Wyoming, which is just north of us, in 2016. And then in 2016, it was the first time we documented it in an adult ponderosa pine. So we think that there might be this expansion westward. And this is, you know, as we started to understand this, we'd always assumed like pinewood nematodes native to the United States is not a problem, right? But then as we started to look at some of the literature, a review had been done on basically pathogenicity assays that were done on different pine species. And so I had mentioned that in the 1980s, there was a lot of studies that had been conducted. The government had funded a lot of this work, the US government, and basically looking at, do we have susceptible species here in the United States? And so on the column on the left, what we see is species that are resistant to pinewood nematode. And what we see on the right are species that are susceptible. And what we find is that a lot of these are exotic pines, like you would expect, mucopine, Austrian pine, Scotch pine, but also here we have ponderosa pine. And so there was some data to suggest that ponderosa pine is susceptible to pinewood nematode. And one thing that's a little frightening about this is that the susceptible host, so this is the distribution of ponderosa pine, basically in the West. And it's pretty extensive and covers a really large range. And we do know that the vectors are here. So we have monocamus species in the West, and there is a distribution of host range. And so really understanding, is the ponderosa, is this a native pathogen that's here the whole time, or is this something that it was native in the East Coast, but it's sort of moving west, has been important. One thing that also makes this even more important is that you start to look at the susceptibility list, and there's a lot of pines here that are in the West that we know are susceptible. So we've got Apache pine, Western white pine, Bishop pine, Monterey, and Southwestern white. So a few years ago, a collaborator and I, Seth Davis, he's a forest entomologist here at CSU as well, we basically got some funding to look at how common is pinewood nematode in our native ponderosa pine forest in the Rocky Mountains. And so Dave Atkins, an amazing grad student who just finished up a couple years ago and started his PhD at University of Wyoming, basically did some field studies to describe the vector abundances and infection frequency across landscapes in the Rocky Mountains, and then identify potential disease epicenters. So is this well distributed across the Front Range in our ponderosa pine forest, or are there pockets of disease that have occurred? So the study was conducted in 2018 and 2019, and we basically had 32 sites that were along the wildland urban interface, which a lot of, these are areas where there's actually like homeowner associations, but they just happen to be sort of next to ponderosa pine forests. And they also included 12 sites that were in urban areas. So 3 in Fort Collins, 3 in Boulder, 3 in Loveland, and then in other areas. And then from each of these sites, what we did was we collected tissue samples. So we selected 10 trees from a lot. We were targeting symptomatic trees from these sites, 10 ponderosa pines. We also then put up traps in the center of the plot to collect insects. So we used host tree volatiles and insect sociochemicals to attract the monocamus beetles. So over time, what we found was that we collected about 280 ponderosa pine samples. And then we had 61 pine samples from urban areas. And then we collected about 5,000 monocamus beetles from our WUI sites, and then 53 from our urban sites. And here's sort of the distribution of those 2 different species that I talked about. So this is captures per site. So this is the number of beetles. And then here's our WUI and urban. The white bars represent Monocamus clamater, and the gray bars represent Monocamus scutulatus. So what you can see from this is that we definitely collected more beetles from our wildland areas, but clamater was more common than scutulatus was, but we were able to collect Monocamus in our urban areas as well. So for screening, identifying if pinewood nematode is in a sample or not can be really tricky if you're trying to do morphology. And so there's been a PCR technique developed called loop-mediated isothermal amplification, or a LAMP. And basically what we're able to do is use this molecular technique, which was developed by a Japanese group and is now sold commercially, to look at whether or not we have a positive. And if you have a positive, they glow in the dark, which is pretty neat. And then if it's negative, it doesn't. So we were able to basically take these wood pieces and these beetles and test them on a molecular scale, so we could do high level testing that can happen much faster. This process takes about an hour or 2. So what we found is that in 2018, we looked at the beetles, so clamater and scutulatus, and then we also looked at our ponderosa pines. And then in 2019, we did not look at the pines, but we looked at urban vectors. And so what we found was that in our pine hosts, ponderosa, the pinewood nematode was found on 3 out of 32 different sites, and it was only found in 10 trees. And we didn't find any pinewood nematode in our urban host, which was pretty interesting. In monocamus, in the beetle, what we found is that the overall infection rate in the WUI sites was about 4.6. And we found more infection in scutulatus than we did in clamater, even though we collected more clamater than we did scutulatus. And we found it very infrequently in our urban sites. So we only found 1 positive of Monocamus scutulatus. So the distribution of pinewood nematode in ponderosa was only found, so it's sort of scattered. Here's a site map. So here's where I am in Fort Collins. And then here's some of these WUI sites. So we only found it in the study in 3 different sites, which are highlighted by the bullseye. And then in these stars, these were areas where my lab had collected it outside of this study. So there were reported cases where people had sent me samples and I was detecting it. And over time, from about 2006 to 2019, what we're seeing is an increase of cases in our exotic pines, which is the dark green, but then also in ponderosa pine in the light green. So it seems like this is something that potentially newly arrived, and then it's starting to spread across the landscape. But when we looked at it in the vectors, what we find is it's pretty much well established in the vectors. So here we've got the crosses here, the red crosses are sites where we found positive vectors, and then the green is where there were no positive vectors. So what you can see is that in 2018, which is the graph on the left, we had 67 different positives with an infection rate of about 3%, 22 sites with more than 1 positive, whereas 10 sites didn't have any positives. And then in 2019, we captured it more frequently at 4.8%. And then we had 30 sites where we found Monochemus infected with pinewood nematode, and only 2 sites where we didn't. So there was actually an increase in the distribution in 2019 compared to 2018. So overall, what we found is that vector abundance across the landscape was really common, infected vectors as well. So we found Monochemus everywhere, which is really common. They're called the pine sawyer beetle. They're attracted to areas where we have fire events. And so they're a really common native species. Interestingly, we do see more in the WUI sites than we do in the urban sites. And this is interesting because we do see pinewood nematode occurring in our urban areas. And so we thought there may be a source of vectors in the urban sites, but we didn't really have results for that. The data didn't represent that. We see Monochemus clamator about 4 times more abundant than Monochemus scutellatus. And then our infection frequency, so ponderosa, there were 3 different WUI sites that had infected ponderosa pine. But over the duration of the study, the pinewood nematode was detected in vectors at every single site. So the nematode is really widely dispersed in our Front Range area, but trees aren't necessarily getting infected at the same distribution. So pinewood nematode is present in our native setting, but it's not uniformly distributed. So we also wanted to identify whether or not we had disease epicenters. And this was some methods that we had taken from basically the human disease literature to look at whether or not we have areas with higher infected insect numbers compared to the rest of the data. So do we have hot spots basically of epicenters or infection? What we found is that we had 4 different epicenters. You can see here highlighted by the bullseye. Interestingly, because of the distribution of how the insects emerge from the host, we basically have 3 which were early season events, and I'll show a picture of this in a second, and then 1 that was later season. But we do have these epicenters that aren't well distributed, and they're pretty close to our urban centers, as you can see here. And so there are hotspots happening, which I think is important to document. One thing that when we did our infection likelihood modeling was that Monochemus scutellatus was 2.5 times more likely to be associated with pinewood nematode than clamator. So even though we have more clamator on the site, we are seeing infection more in scutellatus. And then individuals captured at an epicenter were 20 times more likely to be infected at the beginning of their initial flight emergence than those at peripheral sites, or the ones that weren't those epicenter sites. So a nice description of this. Okay, so this is sort of a busy graph, but basically what we're looking at Monochemus per site. So these were captures. And then the color represents the different species and whether or not it was collected in an epicenter or if it was collected at the peripheral sites. So the solid is epicenters. And so this red solid bar that you see here is Monocamus scutulatus that was collected at epicenters, and then the blue is for clamidor, and then the dots represent peripheral sites, whereas the solids are epicenters. So what you can see is that over time, from July to October, we see a more scutulatus being basically emerging earlier in the year, in the summer. But then later in the summer, we get more clamidor that emerges and at higher levels. Another thing that's pretty easy to identify here is that we have more beetles at epicenter sites than we do at peripheral sites. And this is really interesting because if we overlay the number of infected beetles, so now we've added this dash bar and the solid bar. So the dash bar is the number of positives at epicenter sites, and the dash bar is the number of positives at a peripheral site. And what we see is that beetles that are infected emerge earlier. They're primarily found at these epicenter sites. But this erodes a little bit over time, but we also have another pickup here in late October, which is likely the result of the emergence of Monocamus clamidor. So what we found is that we identified these epicenters, basically that pinewood nematode is not uniformly distributed across the landscape. And so it sort of begs two questions. Is this a newly arrived pathogen in Colorado, right? Is this typical invasive disease that came from Kansas, has moved to the West? Or has it been here? We're just now starting to see anthropogenic disturbances that are driving ponderosa pine susceptibility. So we know that in inoculations that are exotic pines are super susceptible to this pathogen. But we also know that ponderosa pine, if it's doing okay, it's got some tolerance to pinewood nematode, but then if it's stressed, drought stressed, we do see mortality that does occur. Another thing that's really interesting, which has some implications for management, is that these infected beetles potentially emerge earlier than the non-infected. So there'd be a way for us to basically capture the beetles that are infected so that they can't spread to new areas. So what we find is that the risk of our forest, so pinewood nematode disease is the risk of it is really high throughout the Front Range. And higher temperatures have really allowed for the increase in nematode development and disease development. What you can see is that this is temperature from 1985 to 2015 in different areas across the Front Range. And our increases, our temperature is increased in all sites, right? So we're hitting an area where we have vectors on the landscape in pretty high abundances. And these trees, our trees may be more susceptible. And so it's leading to potentially more infections that are happening. Interestingly, though, for our urban sites, is that monocamus, these populations are basically in the WUI sites. And so the WUI sites are disease reservoirs for the urban sites of infected trees. And this is important for trapping if we wanted to trap for the beetles. So we identified these different epicenters. This is important in an area. You might be able to identify these areas that you can do management there. So if we wanted to maybe eradicate or remove the disease pressure radiating from these sites, we can do that. It makes it a little bit more complicated because we do know we have these secondary emergences that occur basically because of clamidor that is out. So it's difficult to manage, but it's possible using the knowledge of these epicenters. Okay, so overall, what we find is that unfortunately, pinewood nematode is in ponderosa in all of our landscapes. So this is a Austrian pine with pinewood nematode in urban landscape. Here we've got one that's semi-urban near Carter Lake. And then here we have one that's right near Rocky Mountain National Park. And so we've hit this stage where pinewood nematode is going to be a disease that we are dealing with both in our urban settings and in our natural ecosystems. We're not clear yet if the factors leading to emergence were really the spread of pinewood nematode into Colorado from basically hopping on these exotic pines as they've spread across Kansas, or if it's been here the whole time. But some of our data really lends to this is potentially an exotic pathogen that's moved in. And this is really being driven, this ponderosa mortality that we're seeing is being driven by climate change. And I like this emerging disease diagram, because what we have basically is emerging diseases that are driven by climate change, which we know are going to increase substantially in our Western forests. We have practices where these exotic pines are basically getting planted into our urban settings, which is allowing for the disease to sort of proliferate and be there. And so we're getting these new shifts in basically host species, vectors, and then the pinewood nematode. So we do know that ponderosa is more tolerant, but as our ponderosa pine has drought and warmer temperatures to basically fight against, it's allowing for more infection to happen. And this is really is a human issue, because along the Front Range, where we have sort of the lowest elevation, people have purchased houses in these areas because of the ability to be next to the forest, but they're dominated by ponderosa pine. And what we're seeing now is mortality happening, where we basically have ecosystem shifts. So our ponderosa pine are turning back into grasslands, and the animal species are starting to move higher up in elevation. And so we've got this relationship between climate change and potentially this introduced pest that is basically causing mortality in our ponderosa pine forest. And it's a big deal. And not even to mention fire, right? So we basically have, this is our drought conditions for the Western United States that was basically taken a couple of days ago. And what you see are just huge pockets of drought that are happening. And it's making our tree species just more susceptible to many things. So whether or not, and this is our wildfire risk potential as well, which is very closely tied to the infection of, or basically the infestation of monocamus species, because they like to have, basically they are attracted to trees that have been damaged by fire. So basically with these two things happening in our Western forest, we are going to see conditions change pretty rapidly. And we're going to see emerging diseases that occur and declines that occur. And so I think this is an interesting factor that we may also be having this pinewood nematode coming in, but that is sort of starting the spiral into disease development and mortality. This will continue to increase, right? So globalization continues, yet our climate conditions are changing faster than our ecosystems can withstand. And I think being here in the Front Range, we're really on the cusp of that. We're seeing ecosystem change happen real time. Our trees are becoming more susceptible to insects and diseases. And I think we have to be really conscious about our urban forests and how they may be increasing risk for invasions that can bridge over into our natural ecosystems. We're not positive this has happened in the pinewood nematode system, but it's an example of one that it may have occurred. As we get stock coming from nurseries, we're planting exotic tree species. We're planting in really stressful environments. We're planting too much of the same host. That is creating environments in our urban settings that then these diseases can move into our ecosystems and our natural ecosystems and be really damaging. So some ways of preventing the spread of invasive species. And we know that our urban forests are super important. You know, I've got this One Health diagram. In the urban environment, having trees can reduce stress and increase human well-being. And so what we want to do is really limit the way that these invasive species are spread. So one is increasing tree diversity in urban areas. If each of you has a homeowner association, if you could be on the tree boards and make sure that our urban areas are planting diversity, it'd be really important. So like I mentioned in Fort Collins, 40% of our trees are either Austrian pines or Scotch pines. And so that's really exacerbated the disease that we're seeing and increased the amount of inoculum. So educating homeowners too of exotic pathogens and species that might be present in their urban areas. Another thing we can do, which is really simple, is basically buy it where you burn it. So you don't want to move firewood around because that can move exotics into different areas. And then lastly, be aware of local quarantines. So one example is emerald ash borer was just introduced into Colorado, and there was a quarantine to move ash wood from where EAB had been found into other areas. And because people weren't aware of it or didn't abide by it, we now have EAB spread into multiple areas into Colorado, which is now threatening our ash. And so this is a trend that I think is going to increase as our Western forests are more stressed out, our urban areas are more stressed out. We're going to get the movement of these invasive pathogens. And I think it's something that we really need to be conscious of and try and limit over time. And just lastly, these are two pictures that I take. This is actually in my neighborhood, and this was in Rocky Mountain National Park. These are ecosystems, both in the urban and the natural ecosystems, that are really important to all of us. And so it's really up to us to try and limit the spread of these potentially invasive pathogens and pests. All right, so if you guys have any questions, let me know. Here's my email if you have other questions that you want to ask or want to reach out to me. Thank you.
Thanks so much, Dr. Stewart. That was very awesome. If anyone has any questions, feel free to write them in the chat, or you can unmute on video and go ahead. If Alyssa allowed that, are they allowed to unmute, Melissa?
So there is one question in the chat if you want to read it, Becky.
Oh, I can say it.
Thank you. Yeah, I've read it, but you can say that.
Yeah. Jane, thanks very much. Lou Gross here. So I'm a bit more familiar with the modeling studies out of Los Alamos, which are not very positive towards long-term pine forests anywhere in the Southwest. Can you say anything? I mean, they're mostly focused, of course, on the individual tree drought impacts. Mm-hmm. Can you say anything about the direct versus indirect impacts of climate on forest declines? And you know, I'm just not familiar what the latest sets of results are.
I mean, in the sense of tree health, drought is going to predispose like the trees to other things. So if you have other secondary pests or pathogens, for sure, on a tree level, that will cause mortality. We are seeing too that some of the mortality we're seeing is just because of drought alone. We cannot associate it with any insect or pest at all. And we've tried, we're looking for these secondaries and we're not seeing anything. So I can't talk long-term, you know, I can't I'm not sure how to capture that long-term or on a large scale, but for sure, drought alone can kill a tree. These other insects and diseases will make that faster to occur. Does that help?
Yeah, but I was also wondering if there's any differences that you see in drought versus pathogen pest response between pinyon or ponderosa or any of the other species.
Yeah, usually the symptoms for the pathogen are pretty distinct. So for drought, we'll just see a couple of branches that start to die back, and then the tree will sort of decline. With the pests and pathogens, we see sort of a similar symptomology with pine nematode, for sure, where you'll have a couple of branches and then the tree sort of declines. But overall, for drought, you'll get basically starvation. And for the pest or pathogen, you might get more uneven distribution of mortality in the tree. Does that make sense?
Yeah, makes a lot of sense.
Yeah.
Yep. Thanks a lot. I'm sorry to see the ponderosas go.
Our ecosystems are changing.
Right. For those of my colleagues here that have never smelled ponderosa, it's it's wonderful.
Yeah. Yeah, so the next, do you want me to read it? So the next question from Ashley is, it sounds like these pathogens pose a threat that might concern real estate, tourism, industries, et cetera. Have these groups served any role in advocating for disease management or climate stewardship? In Fort Collins, or like in Colorado, for sure, this is a huge issue. I mean, we haven't just dealt with like the pinewood nematode, but also bark beetle damage, right? So if you're driving west of the state on I-70, there are huge pockets of trees that have been killed off by bark beetles and other things. There are groups that are advocating, not necessarily for disease management, but for climate stewardship. I think there's, in Colorado, we really recognize that our basically that a lot of these changes are anthropogenic, right? The things that we are doing are causing some of these changes to occur in our environments. And so climate stewardship may be a little bit more. Definitely fire mitigation is more commonly done than disease management. But like going in and preventing forest fire by thinning the forest before they actually occur is now becoming more important and done. But I'm not sure if it's necessary from the tourist groups or from the tourism industries or things like that. But definitely there's recognition of it, I guess. Yeah. Does that help answer the question, Ashley? Okay, thanks.
I have one question, which might be, oh, sorry.
Oh, go ahead.
It might be kind of a little out there based on kind of what you were talking about, but are there any ways to kind of measure stress and immunity in trees? And are people seeing those parameters change with any kind of climate change, anything like
so
that?
you can definitely look at like water potentials. How well is the plant able to move water through the systems? How much water does it have? There's different mechanisms for understanding tree stress, for sure, as it relates to physiological changes. We are seeing differences in tree stress, and you know, we've had droughts happening pretty substantially for the past 20 years, and over these over the time, you're able to see that trees are becoming more stressed, for sure. Yeah, it's a little bit more difficult in pines than it might be in a hardwood, but you can still see it when you examine the trees for the physiological responses. So. So Nathaniel asked, I wonder about possible predators of the pinewood nematode vectors. How can they aid in the control of pinewood nematode prevalence in the landscape? So as far as I'm aware, I don't know of any biocontrols on pine sawyers. Sometimes you might have other insects that feed, but I'm not sure of any close associates. So you might have some biocontrols may be out there, but not, I don't think, at a very high level. We could potentially breed for that or look for entomopathogens that would help control the vector presence. So that is possible for sure that we could look at, but I don't know of any that happen right now in the natural landscape very prevalently and in high densities. Any other questions?
I guess I have one. Is this stimulating any policy or any changes at that level being implemented, at least in Colorado?
No, not really. Unfortunately, I think people for sure were going through limitations of water, right? So from the drought aspect for sure, but not from the pest or pathogen aspect. We really haven't changed. I think one thing that is now being implemented more is increasing species diversity grown in urban forests. Even since, so I've been in Colorado at CSU for about 6 years, and over the past 6 years, there's been a real push away from, we basically had ash trees, honey locust, and our 2 exotic pine species. And if you look around town, that's what's planted. But we're really pushing for species diversity because I think people recognize the importance of the urban environment to the rest of our ecosystems here. You know, most of our cities are so close to our urban environment, to our natural forests. And I think people are starting to understand that. And so I think cities are doing a great job of selling other tree species that aren't going to carry as many pests or pathogens, or we don't know of yet, right? So yeah, I think that has changed quite a bit.
That's excellent, because we always try to push the importance of biodiversity
Yeah,
to health.
exactly. Which it's working, so that's good. And people enjoy seeing the different trees, I think. Yeah.
All right, I think that's right at 1 p.m. here. Again, just a huge thank you to Dr. Stewart. If you do have any other questions and you didn't get her email or anything like that, please just email us at the One Health Initiative and we'll try to relay that. But we really
Thanks so much for the
appreciate it.
opportunity. Yeah, this was fun.